Hello everyone, how are you doing? I hope everyone is doing good. So welcome to another video of inheritance and variation. This is chapter 3 of the Maharashtra textbook and chapter 4 of the CBSE textbook. As we have discussed few topics earlier, you have some basis of inheritance and variation now. In the previous video, we covered the terminologies of genetics. Okay, so you should be knowing the terminologies thoroughly because for understanding the concept further, we will be using those terms very frequently. So you should be aware if I'm talking about what is monohybrid, what is phenotype, what is genotype, you should be aware of the meaning of it. In this video, we are going to see the dihybrid cross, the laws that was derived based on the observation of dihybrid cross, and also laws of dominance and law of segregation. So we are going to see all the laws at the end, and a small topic of backcross and test cross we are going to see in this video. So let's begin the dihybrid cross. Now going back to the monohybrid cross. In monohybrid cross, mono stands for single trait. Yes, mono stands for single trait. So what do you guess by this? Dihybrid means what? Di stands for two. Okay, so there we considered only single characteristics, that is we considered only height. And what were the two variants of the height? Either the plant were tall or the plant were dwarf, or the plants were purple color or the plants were white color. So the dominant we write capital and the recessive allele or the recessive trait we write in small letters. Okay, so dihybrid here we will take two characters, two traits. So we will say we will take the shape of the seed and the color of the seed. Okay, if we would have done only the shape, let's say, then it would be monohybrid cross. But here we are taking two traits, two characteristics of the pea plant, the shape of the seed that has two alternatives, two contrasting traits. Either the shape of the seed will be round or either the shape of the seeds will be wrinkled. And another trait that we are considering is the color of the seed. So yellow color or green color. So yeah, round and yellow are the dominant trait that is seen in the pea plant, and wrinkled and green color seeds are the recessive trait. That is why they are written in small letters and this is written in capital letters. Okay, so and why two? Because we know that for any gene the allele will exist in pairs. Okay, so that is why two capital R, two capital Y for round and yellow, two small r, two small y for wrinkled and green. So dihybrid cross, that is why we are considering two genes at a time. Fine. So it's a cross between two parents differing in two pairs of contrasting characters. Okay, Mendel made some dihybrid crosses in pea plant. Example, so here is the example. He took the yellow round seeds as one parent and the wrinkled and green seeds as another parent. Let's begin. Okay, so in first generation, what will you see? What are you going to see in the first generation? You take the parents. This is how you will represent the parents. You will write the phenotype also and you will write the genotype also. You can mention over here the phenotype is yellow and round and the genotype is 2 capital R, 2 capital Y. You will just write once and you will represent it like this, and a cross has to be mentioned in between. So the gametes we will write separately. Yes, gametes will segregate independently before intermixing or before coming together of the male and the female plant. Let's say this is the female one and this is the male one. Okay? So here what is a possible gamete? Capital R, here capital Y. So because we are considering two traits, so this will be written capital R, capital Y, and here small r, small y. So now these are the gametes of the parents, okay, where we selected two traits. So in first generation, so the first female generation, capital R Y into small r y. What you will get? Capital R, small r, capital R, small r, and capital Y, small y. Okay, so capital R, small r, capital Y, small y. All the progenies that you will see in the F1 will be hybrid, will be heterozygous. No dissimilar allele, right? One is capital, one is small, one is capital, one is small. So this is what you will get in the F1 generation. What is the phenotype? So genotype is capital R, small r, capital Y, small y. And the genotype is obviously the dominant will be expressed. So round and yellow. The seed will be round and yellow. Okay. So this is first generation. What will happen in the second generation? Selfing of the, because all the progenies will have this. So here we write only one. Here we write only one for the ease of understanding. We represent only one, but possibly kitne banenge? 4. Okay. For example, if I write the another pair also over here and the another pair also over here. Okay. So it will be this into this 1. Then this into this 2. Then this into this 3. This into this 4. So 4 possible progeny F1, but all will be same. That is for ease of understanding, we just write once. Same thing, we need not write 4 times. And among them only, we will take one as male, one as female, and we will cross them. Okay. Okay. So in F2 generation, you will get. So we will self all. Basically, the progeny of the F1 will be taken and they will be crossed among themselves. So same genotype and phenotype of the male plant and the same genotype phenotype of the female plant. So let's say one is selected as the male, another is selected as the female. So before carrying out the cross, we will do the gamete separation. Okay, so F2, we will begin with the F2 now. First, we will do the separation of the gametes. So how will you write? So with one capital letter, with the one contrasting trait and the another contrasting trait. So capital R, capital Y, then capital R, small y. Okay, then this capital R with small r. Sorry, this you cannot do. Sorry. Capital R. Capital R is done with both the Y's. Now we will do the small r. Small r with capital Y. And then small r with the small y. So this is how you will get the 4 types of gametes. Okay, I will show you once again. Capital R, small r, capital Y, small y. Okay, how will you do this? How will you represent the gametes? So capital R into capital Y, this is one type of gamete. Then capital R into small y, this is another type. Now this, small r into capital Y, small r into capital Y, and then small r into small y. Okay, small r into small y. So 4 types of gametes you will get for doing the F2 generation. In the same another parent also you will get a similar type. So you can just repeat it like that. Now you will represent this in a checkerboard method. Okay. So 4 on the top horizontally and the 4 gametes vertically. Okay. One representing the male, another representing the female. So like this. So this is how the F2 generation will be represented in a checkerboard method. So these are the 4 gametes represented horizontally of, let's say this is the male gametes and this is the female. Both will be same because we are doing selfing of the F1 generation. So F2 will have the parents same hybrid condition. Now we'll do the crosses. Okay, now we'll do the process as in we'll multiply and let's see what do you get. So the answer is in front of you. So first this into this will give you 2 capital R, 2 capital Y, then this into this, then this into this, then this into this. So first you will multiply this gamete with all the 4, then you will multiply this gamete with all the 4. Okay, and the result you will write in this box. Then this multiplied by this gamete, this multiplied by this gamete, this multiplied by this gamete. So likewise you will do for the remaining. Okay, and this is how the predicted possible progenies will be there for 2 characters. So in monohybrid you had 4 possible progeny in the F2 generation. Here you have how many? 4 here, 4 here, 4 plus 4 plus 4 plus 4. How many? 16. So it got multiplied by the factor of 4. 4 4 are 16 just by increasing one additional character. Okay. So from this now, you will calculate the phenotypic ratio and the genotypic ratio. You can see the gray boxes over here. So the gray boxes are all having the same phenotype, that is yellow and round, 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 yellow and round. So I'm talking about the phenotype, okay? And the green boxes you see, the green boxes are having phenotype as round and green, round and green, round and green. Then you see this light color box, phenotype wrinkled and yellow, wrinkled and yellow, wrinkled and yellow. Okay. And the last one is wrinkled and green. Okay. So phenotypic ratio will be how many? 1, 2, round and yellow are how many? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9. Then round and green are how many? 3 is to 3. Wrinkled and yellow is how many? 3 is to 3. And wrinkled and green is only 1. So all this should add up to how many? Total 16. So 9 plus 1, 10. And 3 plus 3, 6. So 16. So this is the phenotypic ratio. In the similar manner, you will do the genotypic ratio. Okay. So the genotypic ratio, you will write all the types of the genotype of these seeds over here. And you will calculate 2 capital R, 2 capital Y. How many are there? Only 1. So you just write 1 in front of it. 2 capital R, 1 capital Y, and 1 small y. How many are there? So 2. You can see the 2 blue block boxes. Similarly, you will calculate for the rest of the 2, and this is your genotypic ratio. You can either calculate directly, you can manually calculate during the exam, or you can just simply memorize this. 1 is to 2 is to 4 is to 2 is to 1 is to 1 is to 2 is to 1. You can directly memorize this, or you can, once you are done with this process, you can calculate accordingly. Okay. So phenotypic ratio and genotypic ratio of the dihybrid cross. Okay. Now we move to the laws that will come. So what do you see? So what do you get from this? So the first law that we will see over here from monohybrid as well as dihybrid cross is that all the dominant allele is expressed in F1 generation. In F1 generation, you see hybrid condition and the dominant allele is expressed. You do not see wrinkled and green color over here. You see round and yellow color. So this comes up, this tells you that only the dominant allele is expressed in presence of the recessive partner. So law of dominance is observed in F1 generation for both monohybrid and dihybrid cross. Okay, and before coming together, the gametes before coming together of the male and the female, they first separate. They form separate gametes. Okay, that is 1 capital R with 1 capital Y, and then mixed with this. So they first separate before coming together, intermixing with the gamete of the another partner. Okay, another pairing. So that gives you law of segregation. And from F2 generation, we see that no allele is dependent on another. Okay, you see so many varieties. Just by increasing one contrasting trait, you see 4 into 4, 16 possible types of progeny in F2 generation. This is what we call it as independent assortment. So these alleles, they are assorted independently. They are not dependent. So this capital R, capital Y is not dependent or linked or joined to this gamete, or this is not joined to this gamete. All of the 4 are can form different pairs. Yes, of both the parents. In both the cases, they can form different pairs, and they can independently mix with any allele pair of the another parent. For example, this can mix with this, This can mix with this can mix with this. So no two pair of alleles are joined together or are dependent. They are independently and randomly they can mix with any pair of alleles of the another parent. So this gives us third law of Mendel's law of Mendelian genetics, that is independent assortment. So we'll just see the laws. This you have done in your lower standard also. The first law is law of segregation. It states that for any trait, each pair of the alleles will split. As I just told you, they will first segregate. They will split. And one gene passes from each parent to an offspring. Okay. So here 2 capital T, 2 separately you can write, 2 small t you can write. But while passing on to the offspring, only one T will participate from your and another T from the another parent, and you will get a combination of capital T, small t. Okay, like that. So law of segregation basically tells you each pair of allele of the gene will split. So if 2 capital T is the genotype of the parent, so they will split up. They will form individual gametes. And then they can combine with any of the allele of the another parent. This is what is given by law of segregation. Then law of dominance, as this is very simple, only the dominant allele will be expressed. Expression of only one of the form of the parental traits in the hybrid condition. If it is homozygous tall, then obviously tall is the dominant allele, so tall will be expressed. But what happens in hybrid condition, this condition, the dominant allele will be expressed. Okay, so it will display only one of the parent trait, that is the dominant allele. Tallness will be expressed. And then finally, law of independent assortment states that different pairs of alleles are passed on to the offspring independently of each other, as we just discussed with the example. Okay, no 2 allele pairs are dependent or joined or linked to one another. We will see one more concept of linkage, which do not follow Mendel's law. We will see in coming videos. So that we will see in the future videos. But we are talking about Mendelian genetics. So third law of inheritance is law of independent assortment. They can independently combine with any other allele of other parent. Okay, so assortment is nothing but division or segregation like that. So 3 laws of independent assortment. And now we move to the next topic, last topic, that is back cross and test cross. Now, what is back cross? If you see from the figure over here, back cross is, first we'll do the test cross. Test means you want to know the phenotype of the F1 generation. You want to know the genotype. You can see the phenotype by looking, it's observable trait. You can see by the naked eye. And you can determine, okay, this is tall or this is having a purple color flower. But what is the genotype? What is the genotype? Either it is the homozygous condition or it is heterozygous condition. So you want to find out which is the genotype of the F1 generation. So capital P or 2 capital P or 1 capital P, 1 small p. So you saw the phenotype. You know the dominant phenotype has been expressed. But whether this is coming from the homozygous condition or the hybrid condition, so you want to find out that. You want to find out the genotype of the F1 generation. So you will do a test cross. So the name itself is suggesting test cross. You are testing. So what you will do in the test cross? Crossing F1 hybrid. That is what you got in the F1 hybrid. You will take that. And you will do a crossing with its recessive parent. What is a recessive parent? 2 small p. Okay. You will take that plant and you will take the parent plant which has the recessive trait, that is a white flower, 2 small p. Okay. So it is used to find out the unknown genotype of the F1 hybrid. Okay. So Mendel conducted test cross to determine the F2 genotype. You want to know the genotype of the generations, the offspring that will come. Okay. Phenotype, obviously you can see and tell which trait it is, dominant trait or the recessive trait. Okay. Then what is backcross? Backcross is simply again doing the F1 hybrid cross with the any of the parent. So back is basically any of the parent. In test, it is specifically recessive parent. So you have your F1 hybrid over here or F2, whatever you want to test. You want to test the genotype of the first generation, or you want to test the genotype. You want to know the genotype of the F2 generation into recessive parent. Let's say 2 small p. This is your test cross. What is back cross? Same thing. You want to test the genotype of either the F1 generation or F2 generation, but you will do what? You will take any parent. You will take any parent, okay, with any of the parent. Right? So that is another alternative. From that also you will be able to find out the genotype of the F1 generation. You will be able to find out the genotype of the F1 or F2, whatever you are trying to know. So that's all for this video. In the next video, we are going to do incomplete dominance and codominance, which is deviations from the Mendel's law. So up till here, they were following Mendel's law of inheritance. Now we will see certain examples which will not follow the Mendel's law of inheritance, that is dominance, segregation, and independent assortment. So we will start this series from the next video. Okay, make a note of all the things that you are finding it difficult to understand. Take time, keep revising, keep practicing. You have to practice these cross by writing it on paper and memorizing it, because representation will be asked for 4 to 5 marks. So we will solve your doubts in the live class. Till then, take care. Bye bye. Thank you.